The recombinant DNA technology has played a crucial role in the field of medical science and healthcare by helping in early diagnosis and treatment of many diseases. It has also enabled mass production of several effective therapeutic drugs. However, the most striking feature of recombinant therapeutics is that they do not provoke the human body to produce antibodies. On the contrary, use of products isolated from non-human resources can induce an immunological response from the human body. Over the years, biotechnological processes have led to the development of revolutionary medical methods and techniques. These are genetically engineered insulin, gene therapy, and molecular diagnosis. One of the biggest breakthroughs in medical science came with the production of genetically engineered insulin. Insulin is a hormone which regulates the glucose and energy metabolism in the human body. Insulin produced by diabetic patients is either insufficient or impaired. Therefore, they are given doses of insulin at regular time intervals. Earlier, insulin was extracted from the pancreas of slaughtered animals such as pigs and cattle. However, insulin from animal sources often triggered allergies and reactions in some diabetic patients. These problems were overcome with genetically engineered insulin. To understand how it was produced, let us first understand the structure of insulin. Insulin consists of two short polypeptide chains with amino acids, namely chain A and chain B. These chains are linked together by disulfide bonds. In mammals, insulin is synthesized as a prohormone containing an extra stretch known as C-peptide. However, this prohormone is processed before it becomes mature insulin. This made it a big challenge to produce mature insulin without the C-peptide chain from proinsulin. The breakthrough came in 1983, when an American company, Eli Lilly, prepared two DNA sequences that corresponded to chain A and B in the human insulin. Thereafter, they inserted these DNA sequences in E. coli plasmids and insulin chains were formed. Both chain A and B were produced and extracted separately, and later on they were interlinked with disulfide bonds. As a result, human insulin or bioengineered insulin was formed, which is now manufactured and used worldwide. Another innovative application of recombinant DNA technology is gene therapy. It is a collection of methods employed to correct defective genes and cure diseases such as hemophilia and cystic fibrosis. There are a number of steps followed in gene therapy. The therapy begins with the removal of cells from a patient suffering from a genetic disorder. Simultaneously, in the laboratory, a virus, which acts as a vector, is altered so that it cannot reproduce. A therapeutic gene is then inserted into this virus. Next, the altered virus is mixed with the cells taken from the patient. As a result, the cells which were taken from the patient become genetically altered. Finally, the altered cells are injected into the patient. These genetically altered cells produce the desired protein or hormone in the patient, and in this way, gene therapy eliminates the root cause of the disease. Gene therapy was used for the first time in 1990 on a 4-year-old girl suffering from adenosine deaminase deficiency, also known as ADA deficiency. While treating ADA deficiency using gene therapy, lymphocytes are collected from the patient's blood and are then grown in a culture outside the body. After this, a functional ADA, that is, a complementary DNA, is introduced into the lymphocytes with the help of retrovirus vector. Thereafter, the lymphocytes are returned to the patient's blood by transfusion. ADA deficiency is a hereditary disease, and the patients suffering from it bear non-functional T lymphocytes. ADA is caused due to deletion of the gene for adenosine deaminase enzyme, which is essential for the functioning of the immune system. Before the advent of gene therapy, ADA deficiency was cured with the help of bone marrow transplantation and enzyme replacement therapy. Bone marrow transplantation involves the transplantation of blood stem cells derived from bone marrow, while enzyme replacement therapy is a medical treatment that involves the replacement of a certain enzyme by injection. However, none of these methods could cure ADA completely. However, if a functional ADA from bone marrow cells is introduced at an early embryonic stage, a permanent cure can be achieved. However, these cells are not immortal. Hence, usually, patients need regular infusions of genetically engineered lymphocytes. Apart from treatment of diseases, biotechnological methods are also used effectively for molecular diagnosis of diseases. Conventional methods of diagnosis, such as urine and serum analysis, detect the presence of a pathogen at a relatively late stage when pathogen concentration in the body becomes very high. Conversely, methods such as polymerase chain reaction or PCR and enzyme-linked immunosorbent assay or ELISA facilitate early diagnosis. PCR can detect a bacteria or virus even when its concentration is quite low in the body by amplifying their nucleic acid. It is commonly used these days to detect HIV in suspected AIDS patients, genetic disorders, and gene mutations in suspected cancer patients. The other method of molecular diagnosis is enzyme-linked immunosorbent assay or ELISA, which is based on the principle of antibody-antigen interaction. It can detect the pathogenic infection by the presence of antigens such as proteins and glycoproteins, or by detecting the antibodies synthesized against the pathogen. ELISA and PCR have completely modernized the methods of disease detection. Along with gene therapy and genetically engineered insulin, these biotechnological innovations have transformed the face of medicine and healthcare.